Welcome back to the Deep Dive. Today we're tackling something I find genuinely fascinating, almost a paradox, in the world of molecular diagnostics.
Oh, what's that?
Well, we're in this era where sequencing a person's entire genome is getting faster, cheaper.
Mm,
You hear about it all the time.
right. NGS, whole genome sequencing, it's definitely center stage.
Exactly. But that leads to a really critical question. How do scientists, how do pathologists actually find the massive structural changes in DNA? I mean, the really big stuff, deletions, inversions, bits of chromosomes swapped around.
The things that are maybe too large or too architecturally complex for standard sequencing to reliably pick up.
Precisely. Those huge alterations that NGS might, struggle with or miss entirely.
It's a perfect topic for a deep dive, actually, and it really speaks to how different technologies have to work together. The source material you shared was clear on this point.
Yeah, it highlighted cutting-edge stuff, but kept coming back to the value of, quote, older technologies.
Exactly. While next-generation sequencing grabs the headlines, there are absolutely crucial genetic problems, these large structural variants, that sequence-based methods just aren't ideally suited to detect.
So what's the plan?
So our mission today is to really get into one of the most important of those foundational visual techniques, fluorescence in situ hybridization. You know it as FISH.
Fish. Okay.
We're going to dissect its mechanism, which is surprisingly elegant, explore its really critical clinical uses, especially in cancer diagnostics, and understand why this technique, which dates back decades, is still absolutely indispensable.
Okay, let's unpack this a bit then, because, like I said, when most people think about looking at DNA, they probably picture, reading that stream of A, T, C, G, S.
Looking for the typos.
Right, the single letter changes, the small insertions or deletions that NGS is brilliant at finding. But you're saying, and the source material emphasizes, that in many diseases, especially things like complex congenital syndromes or aggressive cancers, the problem isn't a typo.
It's more like an architectural failure, a blueprint error.
Yeah. Like, what if it's not just a misspelled word, but a whole sentence, maybe even a huge paragraph? I think the source mentioned sizes up to 200,000 letters.
That's right, 200 kilobases.
What if that much DNA has been just deleted or duplicated or picked up and moved to a completely different chapter in the genome?
Well, that is the kind of structural variation, the large genomic alteration that defines many cytogenetic disorders. And if you rely only on sequencing approaches that chop the DNA into tiny pieces first.
The short reads.
Exactly, the short reads. Well, they inherently struggle to map correctly across those massive breakpoints, or through the repetitive DNA sequences that often flank these structural changes. There are technical limitations.
So it's hard to piece the puzzle back together correctly if some of the pieces are huge or moved around?
That's a good way to put it. And that's precisely why hybridization methods like FASH still provide this incredibly valuable visual roadmap. You're literally seeing the structure on the chromosome. It's essential when the problem is architectural, not just sequential.
It still kind of blows my mind a little that in this age of super fast sequencers, we're still relying on a process that involves, heating up DNA and using glowing dyes. Walk me through how it actually works. Why hasn't this been, engineered out?
I think to understand why it persists, you have to appreciate its fundamental elegance and power. Fluorescence in situ hybridization. It's a hybridization-based technique, first and foremost.
Okay.
And the name, often happens in science, really tells you everything. Fluorescence, well, that's the glowing dyes we use to see things.
Makes sense.
In situ, that's just Latin for in the original place.
Ah, so you're looking at the DNA right where it lives, inside the cell.
Exactly. Inside the nucleus of an intact cell, or at least a preserved one, we're visualizing the DNA in its natural context, more or less.
Okay, so the key isn't just reading the sequence letters, it's physically locating a specific sequence. And you mentioned the whole test hinges on a key component. The probe.
Absolutely. The probe is everything.
These aren't just random bits of DNA, are they? What makes a FISH probe so special?
Right. It's highly specific.
Yeah.
The probe is essentially your targeted searchlight. It's a fragment of DNA, usually cloned, that's been synthesized to be perfectly complementary to the one specific genomic sequence you're trying to find.
Complementary meaning it will stick to it, like Velcro.
Precisely. A sticks to T, C sticks to G. The probe is designed to match a long stretch of the target sequence perfectly. And crucially, as the source material highlights, these are typically large fragments. We're talking up to 200 kilobases or 200,000 base pairs long.
Wow, 200 kilobands. That's pretty big compared to a gene. Why that specific size? Is it arbitrary or is there a reason?
Oh, there's definitely a reason. It's sort of the engineered sweet spot.
Sweet spot, how so?
Well, think about it. If the probe were too small, say only 5 or 10 kilobases, the fluorescent signal you attach to it would be too weak, too dim, and maybe too diffuse to see reliably under the microscope. You wouldn't get a clear dot.
Okay, need enough signal?
Right, but if you made it significantly larger, like several million base pairs, a few megabases, it becomes technically very challenging and extremely expensive to synthesize accurately, to clone it, and to label it effectively with the fluorescent dyes.
Diminishing returns and technical hurdles.
Exactly. So that 100 to 200 kilobatter range, roughly, gives you two critical things. First, extremely high specificity. It's long enough that it's virtually guaranteed to bind only to its intended target sequence in the entire genome.
Low chance of sticking to the wrong place.
Vanishingly low. And second, it provides enough physical length, enough DNA real estate to attach sufficient fluorescent molecules to create a bright, distinct punctate signal that's easy to see.
Okay, so we have this highly specific, brightly glowing homing beacon, this 200 kilobatter DNA probe, tagged with maybe a red dye or a green dye.
Yeah.
Now, how does it actually find and stick to that one sequence inside a cell nucleus? That sounds complicated. The DNA in there is all tangled up, isn't it?
Yeah.
And double-stranded.
It is, and that's where the in situ preparation part comes in. You have to overcome the natural state of DNA. As you said, genomic DNA is normally a double helix. The two strands are wound around each other and tightly paired up by hydrogen bonds.
Right.
The probe, which we typically prepare as double-stranded too, needs to find and bind to just one of the strands of the target DNA in the cell.
So you somehow have to unzip the target DNA first, pull the strands apart.
That's exactly it. We have to achieve denaturation, or what scientists often call melting the DNA.
Melting, like with heat.
Primarily, yes. The cell sample, which could be cell spread on a microscope slide, is subjected to heat, usually around 95 degrees Celsius, often along with some chemical pretreatments as well.
95°, that's almost boiling.
It is. And that high temperature provides enough energy to break those relatively weak hydrogen bonds holding the two strands of the double helix together. So effectively, you temporarily turn the double-stranded DNA in the nucleus into single-stranded DNA.
Okay, but if you've just blasted it with heat and pulled everything apart, doesn't that make the whole thing really unstable? How do you stop the original two strands from just snapping back together before your probe even gets a chance to find its spot?
That is a critical technical challenge, and it demands very precise temperature control. It's not just heat and walk away.
Right.
After that initial high-temperature denaturation step, the temperature is carefully and slowly lowered, usually down into the mid-50s to low 60s Celsius range. And it's held there for quite a while, several hours, sometimes even overnight. The slow, controlled cooling phase is the hybridization phase.
Okay, so cooler but still warm.
Exactly. And during this hybridization period, we introduce the labeled probe into the sample mixture. Now, the probe is typically present in a much higher concentration than the target sequence within the cell.
So it has an advantage.
It has a numerical advantage, yes. And because it's designed to be perfectly complementary to its target sequence, it effectively competes with the original genomic strands to bind to that now exposed single-stranded target DNA. Given enough time and the right conditions, the probe finds and hybridizes binds strongly to its specific spot.
And because it's got that fluorescent tag.
The result is that the specific chromosomal region the probe was designed to find is now labeled with a bright fluorescent color. You put the slide under a special fluorescence microscope, use the right filters, and you can see these distinct glowing dots.
And you mentioned this can be done on different types of cells or different states of the cell, metaphase chromosomes versus interphase nuclei. What's the difference there, and why does that matter clinically?
Yes. That choice really speaks to a classic trade-off in diagnostics. Speed versus resolution.
Okay.
So metaphase preparations use cells that have been caught right in the middle of dividing. Their chromosomes condense into those familiar X shapes and line up.
The classic picture from biology textbooks.
Exactly. If you apply FESH probes to these perfectly condensed metaphase chromosomes, you get the highest possible spatial resolution. You can see exactly which chromosome the signal is on and even which part of the chromosome arm. You can see if a large chunk has moved.
So metaphase FESH is like looking at a perfectly organized library bookshelf where you can see exactly which book, which chapter the glowing sticker is on.
That's a great analogy. Highest detail. However, preparing a good metaphase spread requires culturing the cells first, stimulating them to divide. And that takes time, usually 24 to 72 hours, sometimes longer.
So not instant.
Not at all. Interphase FESH, on the other hand, Uses cells that are just in their normal, non-dividing resting state. We call it interphase. In these cells, the chromosomes aren't condensed, they're decondensed and kind of tangled up within the nucleus, like spaghetti.
So the library books are all just piled on the floor.
Kind of, yeah. The spatial resolution is much lower. You can't easily tell which chromosome a signal is on just by looking. But the preparation is almost instantaneous. You can take cells directly from a blood sample or a biopsy, put them on a slide, and perform FIH right away.
Okay, so interphase is faster, but fuzzier, less detailed. Why would you choose the faster, fuzzier option then?
Speed.
Hmm.
In many clinical situations, especially in cancer diagnostics, speed is absolutely critical.
Right.
Think about analyzing a fresh tumor biopsy. You can make a touch prep, just touch the tissue to a slide to get some cells, fix them immediately, and run interphase FIH within hours, sometimes even minutes for very urgent cases. You lose the high-resolution mapping you get with metaphase, but you can still do crucial things, like simply count the number of signals per nucleus.
Okay. So even if the books are piled up, you can still count how many copies of a specific book there are.
Exactly. If you're looking for something like Down syndrome trisomy 21, you use a probe for chromosome 21.
Mm-hmm.
In a normal interphase nucleus, you expect to see 2 signals, 2 dots. If you consistently see 3 dots.
You've got visual confirmation of the trisomy instantly.
Instantly, without waiting potentially 3 days for a full metaphase karyotype culture, it turns an abstract genetic question into a concrete, countable, visual answer for the pathologist, often providing critical information much faster. #22. Detailed clinical applications and sample flexibility.
That speed, coupled with the visual confirmation.
Yeah, I can see why that's so powerful. And this is where the versatility you mentioned really comes into play, right? Connecting the how it works details to the actual clinical impact. The source material really emphasized the flexibility that FAHASH can be done on almost any kind of sample you can think of.
It's quite remarkable, really. You can use it on prenatal samples like amniotic fluid cells or chorionic villus samples,
Okay.
peripheral blood lymphocytes, obviously, bone marrow aspirates for leukemia diagnosis, those touch preparations from fresh tumor biopsies we just talked about,
Yeah.
but also, and this is incredibly useful, on formalin-fixed paraffin-embedded tissue sections, the kind routinely stored in pathology labs. Wait, so you can go back to old samples from years ago?
Absolutely. That ability to use fixed archival tissue is huge for research. It allows scientists to conduct retrospective studies, take tumor samples collected maybe 10, 20 years ago, perform FIH for a specific gene alteration, and correlate that finding with the patient's long-term outcomes. It's foundational for understanding how specific genetic changes drive cancer progression over time.
Wow, okay. But for the day-to-day clinical world, what are the main types of questions FIH is used to answer? You mentioned counting signals.
Right. For clinical diagnostics, FIH is primarily valuable for rapidly and accurately answering, I'd say, 4 main types of crucial questions, and often different types of probes are designed for each application.
Okay, let's tick through them. Application 1.
Application 1, detecting aneuploidy. We touched on this. Aneuploidy just means having an abnormal number of chromosomes, typically gaining or losing entire chromosomes. Think trisomy 21, Down syndrome, trisomy 18, Edwards syndrome, trisomy 13, Patau syndrome, or abnormalities in sex chromosomes like Turner or Klinefelter syndromes.
So conditions caused by having, say, 3 copies of a chromosome instead of the usual 2, or maybe just 1 copy.
Exactly. Now, traditional karyotyping, looking at the metaphase chromosomes, can see these whole chromosome changes. But FIH, especially interphase FIH, provides that rapid, targeted, visual confirmation.
Give me another example where that speed is critical, beyond prenatal.
Well, sometimes in newborns with multiple congenital anomalies, there's an urgent need to know if a common trisomy is present to guide immediate medical management. Or in certain leukemias or lymphomas, specific numerical abnormalities can have prognostic significance or influence treatment choices. FIH can give you that answer quickly using probes that target the centromeres, the highly repetitive DNA at the center of each chromosome.
Centromeric probes. So they just stick to the middle of a specific chromosome.
Right. Use a probe labeled, say, green for the chromosome 18 centromere and red for the chromosome 21 centromere. You look at the interphase nuclei, you count the green dots and the red dots, 2 of each, normal count. 3 red dots, likely trisomy 21. It's straightforward counting.
Okay, so that handles whole chromosome gains or losses. But FIH is also key for problems that are actually too small to be seen reliably by that standard karyotyping, right? Application 2, subtle microdeletions.
Yes, this is a really important niche for FEFISH. We're talking about losing a small piece of a chromosome, a deletion, but one that's too small to be resolved by just looking at the banding pattern on a metaphase chromosome.
How small are we talking?
Traditional G-banded karyotyping has a resolution limit of maybe 5 to 10 megabases. That's 5 to 10 million base pairs. Anything smaller than that is usually invisible.
Wow, okay.
But many well-known genetic syndromes are caused by deletions much smaller than that, often in the range of just 50 kilobases, up to maybe a few megabases. Think of syndromes like DeGeorge syndrome, 22q11.2 deletion, Williams syndrome, 7q11.23 deletion, Prader-Willi-Engelmann syndromes, 15q11q13 deletions. These are often called microdeletion syndromes.
And that's where that 200 kilobase probe size we discussed earlier becomes really critical if it's right in that detection gap.
It fits perfectly in that gap. It's too small for karyotyping to see, but often these deletions are large enough or flanked by repetitive DNA, such that they can sometimes be tricky for standard short-read sequencing to characterize confidently, especially confirming the exact breakpoints. PEFISH fills this gap beautifully.
So if a child presents with developmental delay, maybe some characteristic facial features, and the routine chromosome analysis, the karyotype, comes back looking normal, FIH might be the next step.
Absolutely. For these known microdeletion syndromes, we use a specific type of probe called a locus-specific identifier, or LSI probe. It's designed to bind directly to that critical region known to be deleted in the syndrome.
Okay.
You typically use that specific probe, say labeled red, along with the control probe for a different region on the same chromosome, say labeled green, as an internal reference. In a normal individual, you'd expect to see 2 red signals and 2 green signals in each nucleus.
2 copies of each region.
Right. But in a patient with the microdeletion, you'll see only 1 red signal because 1 copy of the target region is gone, but still 2 green signals. The control region is intact.
Ah, so the absence of 1 red dot is the diagnostic signal.
Precisely. It provides definitive visual confirmation of that specific microdeletion. It makes an otherwise invisible genetic loss completely obvious under the microscope, transforms it into a clear yes-no answer.
That's powerful. Okay, application 3.
Application 3. Detecting complex translocations. This is where FIH is absolutely essential, particularly in cancer, especially leukemias and lymphomas. A translocation is when pieces of 2 different chromosomes break off and swap places.
So part of chromosome 9 ends up on chromosome 22, and part of 22 ends up on 9, for example.
Exactly. That's the classic example of the Philadelphia chromosome seen in chronic myeloid leukemia, CML. Now, routine karyotyping can often see that a translocation has occurred, especially if the swapped pieces are large or have different banding patterns. But sometimes the exchange is subtle, or the chromosomes involved look very similar, or the quality of the metaphase preparation isn't perfect. Karyotyping might miss it or struggle to identify the exact partners involved.
And FISH can nail it down. How?
Using specifically designed probe strategies, there are 2 main types here: fusion probes and break-apart probes.
Okay, fusion probes first.
For a fusion strategy, you use 2 probes, each labeled with a different color, designed to bind to sequences on the 2 different chromosomes that are known to be involved in a specific translocation.
Mhm.
Let's take that CML example. You'd use a probe, say red, for the ADL1 gene region on chromosome 9, and another probe, say green, for the BCR gene region on chromosome 22. In a normal cell, you'd see separate red and green signals. But if the Philadelphia translocation has occurred, the BCR and ABL1 genes are brought right next to each other, fused together on the derivative chromosome 22.
So the red and green probes will be right on top of each other.
Exactly. And when those red and green fluorescent signals overlap spatially, they merge optically to create a third, distinct color, typically yellow or orange, depending on the dyes. Seeing that fusion signal is a direct visual confirmation of the translocation.
Very clever. What about break-apart probes?
Break-apart probes work differently. Here you use 2 differently colored probes, say red and green again, that are designed to bind to sequences that flank, or sit on either side of, a known breakpoint within a single gene that's often involved in translocations.
Okay, so they normally sit right next to each other.
Right. In a normal cell, because the probes bind close together on the intact chromosome, you see a merged yellow-orange signal, or just 2 very closely spaced red and green signals. But if a translocation occurs, that breaks the chromosome between where those 2 probes bind.
Ah, the probes get separated onto the 2 different derivative chromosomes.
Precisely. The fusion signal breaks apart. You'll now see 1 separate red signal and 1 separate green signal. Seeing them split apart tells you that gene has been disrupted by a translocation, even if you don't know what the partner chromosome is yet. This is very useful for identifying rearrangements involving genes like MYC or BCL2 or MALT1 in lymphomas.
Okay, that makes sense. Now, you mentioned speed being critical. Is there a translocation scenario where a fish's speed is absolutely life or death important?
There absolutely is, and it's probably the most dramatic example of fish providing immediately actionable intelligence. Acute promyelocytic leukemia, or APL.
APL. Okay, what's the story there?
APL is a subtype of acute myeloid leukemia, and it's typically caused by a very specific translocation between chromosome 15 and chromosome 17. This translocation, T1517, fuses the PML gene on chromosome 15 with the retinoic acid receptor alpha gene, RRRA, on chromosome 17.
Okay, the PML-RRA fusion. Why is that so critical to find quickly?
Because this specific fusion protein profoundly disrupts normal blood cell development, but incredibly, it's uniquely sensitive to treatment with a vitamin A derivative called all-trans retinoic acid, or ATRA.
Retinoic acid, like in skin creams.
Related, yes, but used therapeutically here. If a patient presents with symptoms suggesting acute leukemia and the initial blood smear looks like it might be APL, the hematologist and pathologist are under immense pressure. They need to confirm that specific T1517 translocation as fast as humanly possible. We're talking hours.
Why the urgency?
Because if the PML-RRA translocation is present, starting ATRA treatment immediately is incredibly effective. It can induce remission rapidly.
Yes.
But if that specific translocation is not present, if it's a different type of leukemia, ATRA is completely useless, and giving it just delays starting the necessary different chemotherapy, which is dangerous.
Wow. So the treatment decision hinges entirely on detecting that one specific translocation fast.
Entirely. And FISH, using either fusion probes for PML and aware RRA or break-apart probes for RRA, is the fastest, most reliable way to get that definitive answer, often within 2-4 hours of the bone marrow sample arriving in the lab. Seeing those fused yellow signals or seeing the RRA probes split apart directly dictates immediate, potentially life-saving therapy. It bypasses the days it might take for full karyotyping or complex sequencing confirmation.
That is, yeah. That's a powerful example of actionable intelligence. Okay, one more main application.
The fourth major application, particularly crucial in solid tumor oncology, is detecting gene amplification.
Amplification meaning making lots and lots of extra copies of a specific gene.
Exactly. Instead of the normal 2 copies per cell, the cancer cell might make 10, 20, 50, even hundreds of extra copies of a particular gene. This leads to massive overexpression of the protein that gene codes for, which often drives tumor growth and aggressiveness.
And if can visualize this, how does that look different from just detecting presence or absence?
It's quantitative. You're essentially counting the signals again, but looking for a large excess. The classic textbook example here is the amplification of the HER2 gene in breast cancer.
HER2, right. That's linked to targeted therapies.
Precisely. The gene's official name is ERBB2, but it's commonly known as HER. To test for its amplification using FISH-ARCH, pathologists typically use a dual probe strategy. One probe, say labeled red, targets the HER2 gene itself on chromosome 17. The second probe, labeled green, targets the centromere of chromosome 17, often called the CEP17 probe. This green probe acts as an internal control for the chromosome copy number in that cell.
Okay, so you compare the number of red dots to the green dots.
Exactly. In each cancer cell nucleus, you carefully count the number of red HER2 signals and the number of green CEP17 signals. In a normal cell, or a tumor cell without amplification, you'd expect roughly the same number of red and green signals, maybe 2 of each, giving a ratio close to 1.0.
But if HER2 is amplified?
You'll see way more red signals than green signals. You might see, say, 15 red dots, but only 2 or 3 green dots. The ratio of HER2 signals to CEP17 signals becomes high, maybe 5 or 10 or even higher. A ratio above a certain threshold, usually 2.0, is defined as amplification.
And that ratio is the key piece of information for treatment.
It absolutely dictates treatment. If the FISH test shows HER2 amplification, the patient is eligible for therapies that specifically target the HER2 protein, like trastuzumab, Herceptin, or pertuzumab. If it's not amplified, those drugs won't work and shouldn't be given.
So FISH here provides the definitive yes-no for that targeted therapy. Are there other key examples?
Oh, yes. Another critical one is amplification of the NMYC gene, sometimes just called MYCN, in neuroblastoma, which is a common childhood cancer. High-level NMYC amplification seen by FEHH is a very strong indicator of aggressive disease and poor prognosis, requiring much more intensive treatment protocols. So again, FEOSH provides critical prognostic and therapeutic information just by counting those dots. It turns a quantitative genomic change into a clear clinical decision, visualized right there on the slide.
Okay, we've built a strong case for retorsh being fast, visual, highly targeted, and clinically crucial for aneuploidy, microdeletions, translocations, and amplification. But let's zoom out again to that bigger picture, the overall diagnostic landscape. Especially with sequencing getting cheaper and seemingly more comprehensive, we have to come back to that technical limitation you mentioned right at the start. Why exactly are these large genomic alterations often missed or poorly characterized by standard sequencing?
Right, and it really is the fundamental reason why FEOSH and hybridization-based methods in general remain so important. It comes down to how most next-generation sequencing NGS works.
The short reads again.
The short reads. The DNA is first broken into millions of small fragments, typically only 150 to 300 base pairs long. These short fragments are then sequenced. The challenge comes afterward in the bioinformatics step, where computer algorithms try to align these millions of short reads back to a standard reference genome map.
Like putting together a shredded document.
Kind of, yeah. Now, if the change is small, like a single letter typo, a point mutation, or a tiny insertion or deletion, the short reads usually align pretty well, and the software can spot the difference compared to the reference.
Okay.
But now imagine you have a massive structural rearrangement. Let's say a 200 kilobase piece of chromosome 5 has been deleted entirely. Or maybe a huge chunk of chromosome 8 has flipped itself around, an inversion. Or two large pieces from different chromosomes have swapped places, a translocation.
What happens with the short reads then?
The alignment algorithms really struggle. For a large deletion, the software might notice a region where suddenly far fewer reads align than expected.
A drop in coverage.
Exactly, a drop in read depth or coverage. That suggests a deletion, but it's often hard to be certain about the exact boundaries, the breakpoints, just from that drop. And crucially, the short reads can't span the deletion to physically link the sequences on either side of the gap.
They're too short to jump across the missing piece.
Precisely. Similarly, for translocations or inversions, the reads that cross the breakpoint might align to two completely different places in the reference genome, or align in the wrong orientation. This creates ambiguous or conflicting mapping signals that can be very difficult for algorithms to interpret reliably, especially if the breakpoints occur within repetitive DNA sequences, which are notoriously hard to map uniquely.
So the algorithm might flag something weird is happening, but it can't give you a definitive picture of the new structure.
Often, yes. It loses the overall architectural context. Short-read sequencing is phenomenal for reading the text, but it's inherently limited in its ability to confirm the large-scale chapter organization. It's trying to understand the blueprint by looking only at individual bricks. That's why hybridization methods like FISH, which physically label and visualize those large regions, provide a level of structural certainty that sequencing often lacks in the specific area.
Okay, that makes perfect sense why it's still needed. But
Yeah.
this brings us directly to what's often cited as FISH's main weakness, doesn't it? The reason people sometimes push towards arrays or sequencing, FIH is incredibly sensitive, but only if you know exactly what you're looking for beforehand.
That is absolutely its primary limitation. You must have prior knowledge, or at least a very strong suspicion, of the specific chromosomal region or gene you think is altered.
You need to know which probe to order.
Precisely. You have to design or select that 200 kilobauer homing beacon probe to target a specific address. You can't just take an unknown tumor sample, run a generic FIH test, and ask the broad question, what structural abnormalities are present? It doesn't work like that.
You have to ask a specific question like, is the HER2 gene amplified? Or is the PMLR translocation present? Or is the DeGeorge region deleted?
Exactly. It's a hypothesis-driven technique, and that's a significant drawback, especially in situations like diagnosing a child with developmental delay of unknown cause or characterizing a cancer where you don't know the specific driving mutations yet.
So if T-SHASH is the highly targeted, super bright searchlight, what's the alternative for casting a wider net? What's the wide-angle surveillance camera in this analogy?
That role is primarily filled by cytogenomic array technologies. You might hear terms like array comparative genomic hybridization, array CGH, or SNP arrays, single nucleotide polymorphism arrays.
Okay, arrays. How do they work differently?
Instead of using one or two specific probes on a cell, arrays use hundreds of thousands or even millions of tiny DNA probes spotted onto a small glass chip. These probes represent sequences spread across the entire genome. You then take the patient's DNA, label it with one fluorescent color, say green, and a normal reference DNA sample, label it with another color, say red. You mix them together and hybridize them to the array chip.
So you're comparing the patient's DNA to a normal control across the whole genome at once.
Exactly. A computer scans the chip and measures the ratio of green to red fluorescence at each probe location. If the patient has a deletion in a certain region, there will be less green signal binding there compared to red, and the software flags it as a loss. If they have a duplication, there will be more green signal flagged as a gain.
So arrays give you a global survey of copy number changes, gains and losses across the entire genome without needing to know where to look first.
Precisely. That's why arrays have become the first-line test for investigating unexplained developmental delays, intellectual disability, or multiple congenital anomalies in children. They can find unexpected deletions or duplications anywhere.
Okay, so arrays seem great for discovery of unknown, unbalanced changes. But what can't arrays do that FCS still excels at?
This is a critical distinction. Array CGH fundamentally works by detecting changes in the amount of DNA, the copy number, or dosage, if a structural rearrangement is balanced.
Meaning pieces were swapped, but no significant amount of genetic material was actually gained or lost overall.
Exactly. Like many of the translocations we see in leukemia, such as the PML-RRA fusion in APL or the BCR-ABL fusion in CML, two chromosome swap pieces, the genes get fused into a new oncogene, but the total amount of DNA in the cell remains essentially normal.
So the array wouldn't see it? The red-green ratio would look normal?
The array is completely blind to balanced translocations. It also struggles with low-level mosaicism, where only a fraction of cells have the abnormality, and sometimes with precisely mapping breakpoints in complex rearrangements. And it can't tell you the orientation of duplicated segments or where inserted material came from.
So FESIsh, using those fusion or break-apart probes, remains the gold standard for detecting those critical balance translocations that drive diseases like leukemia, which arrays would miss entirely.
That's absolutely right. It highlights why neither technology can fully replace the other right now. They have complementary strengths and weaknesses.
So you end up using them together, or
Yeah.
sequentially.
Often, yes. It creates a more complete diagnostic picture. For instance, in pediatric genetics, an array might be the first test to find an unknown deletion or duplication. FISH might then be used to confirm the finding or to test the parents to see if it's inherited or de novo. In cancer, sequencing or an array might reveal a complex pattern of gains and losses, but FISH might be needed to confirm a specific amplification like HER2 or to quickly detect a known translocation like PML-RRA for immediate treatment decisions.
Okay, that synergy makes sense. Let's talk about the practical side too, cost and time. We hear sequencing is getting super cheap, maybe under $1,000 for a whole genome soon. Doesn't that make HESH look expensive or slow by comparison?
It's a nuanced comparison. Yes, the raw cost of sequencing per base has plummeted dramatically, but analyzing and interpreting a whole genome sequence, especially looking for complex structural variants, still requires significant bioinformatics expertise, computational power, and pathologist time. Getting a clinically validated report back from WGS, particularly for structural changes, can still take days or even weeks in many settings.
The analysis bottleneck.
It's a real bottleneck. FHH, on the other hand, while requiring specific probes which have development costs, is relatively inexpensive per targeted test once established. More importantly, the interpretation is often immediate and visual. A trained cytogeneticist or pathologist can look at the slide under the microscope, count the signals, and make the call often within hours for urgent cases, as we discussed with APL.
So faster turnaround time for specific, actionable results.
Much faster in many critical scenarios, and that visual certainty, actually seeing the amplification or the translocation, provides a high degree of confidence. So despite the rise of NGS, the source material is right. Older technologies, particularly FHH, continue to play a vital role. For many complex cytogenetic disorders, and especially for rapid, targeted diagnoses where speed is paramount, the combination of traditional karyotyping, FISH, and sometimes array technology remains the practical gold standard. Newer tech doesn't automatically replace foundational methods that still do certain jobs better or faster.
It sounds like the diagnostic world doesn't see FSH as, a dusty old tool, but more like that indispensable specialized instrument you need for certain critical tasks. The one that finds the problems hiding in plain sight, but only if you know exactly where and how to shine that specific fluorescent light.
That's a very good way to put it. It's a targeted spotlight with unique capabilities. Hashtag tag4, conclusion, and a final thought.
So let's try and synthesize this. What does this deep dive into fish age really tell us about the modern molecular diagnostic toolkit? We've seen it's not just some holdover technique. It remains this incredibly powerful visual method for finding those big structural genomic changes.
The aneuploidies, the microdeletions, the crucial translocations, the gene amplifications.
Right. The very things that purely sequence-based approaches can sometimes struggle with or miss entirely because of their sheer size or complexity. FSH takes what could be confusing data from an algorithm and transforms it into clear, visible, actionable clinical information right there on the microscope slide.
And if we boil down its function, FSH works by harnessing that fundamental principle of DNA hybridization. It uses these carefully designed, fluorescently tagged DNA probes, often quite large, around 200 kilobammets, to physically find and light up very specific sequences on the DNA that's been gently denatured inside the cell nucleus.
Allowing direct visualization.
Exactly. This targeted visual approach lets pathologists rapidly and accurately diagnose devastating diseases like acute promyelocytic leukemia based on seeing a specific translocation, or characterize a breast tumor by quantifying HER2 gene amplification, directly guiding life-saving treatment decisions where speed and absolute certainty are critical.
It really is a remarkable example of merging molecular precision with the power of just looking and seeing.
It is, and that actually leads us perfectly into a final, maybe provocative thought for you, the listener, to consider.
Okay, let's hear it.
We spent a good amount of time discussing FSH's main limitation, the fact that you need prior knowledge. You have to know which specific gene or region to target with your probe.
Right. Hypothesis-driven.
But looking towards the future, whole genome sequencing, WGS, which aims to sequence everything, is constantly improving. The technology and the bioinformatics algorithms are getting better and better at detecting novel structural rearrangements, finding deletions, duplications, inversions, translocations that we didn't even know to look for without needing a specific probe.
So WGS is aiming to become the tool that can find the unknown structural changes too.
That's the goal. Now, today, as we discussed, WGS still faces challenges in reliably calling all types of large structural variants, especially balanced ones, and the interpretation pipeline can still be complex and slow for these findings. But let's project out, say, 10 years.
Okay.
Imagine a future where WGS truly costs pennies, where the artificial intelligence or machine learning needed to instantly and accurately interpret all forms of structural variation from that sequence data becomes routine and completely reliable. In that future, what will be the truly indispensable protected niche for a targeted visual method like FSH? Will its role shrink purely to those ultra-rapid emergency diagnostic scenarios like the APL case where even a few hours saved matters immensely? Or will there always be an inherent value in a mutable place for the simplicity, the reliability, and the absolute visual certainty that comes from actually seeing that glowing 200 kilobauer probe physically stuck to the chromosome or confirming its absence?
That question of how visual certainty will ultimately integrate with, or perhaps be superseded by, comprehensive algorithmic detection.
Yeah. That convergence, or maybe continued divergence, is really the next frontier for molecular pathology to navigate. What role remains when the machines get really good at seeing everything? It's something to definitely keep an eye on.